Hello, everyone, and welcome to today's National Soil Survey Center webinar. My name is Sean McVeigh, National Training Coordinator for the Soil Science Division, and host for today's webinar, Corn stover removal reduces soil microbial biomass and alters microbial community structure under irrigated continuous corn in eastern Nebraska. This webinar is being recorded, and all participants join the webinar in listen-only mode. You receive the webinar audio through your device's speakers. There is no telephone dial-in. If you're having audio difficulties, please check the various ways your computer speakers may be muted or have their volume set low, including the speaker adjustments available in the Adobe Connect interface. You can maximize your webinar experience in Adobe Connect by shutting down VPN and any other programs that might compete for bandwidth. This includes email and Microsoft Outlook and instant messaging in Skype. Taking a look at our webinar room layout, Adobe Connect has content pods that include the feature presentation, chat, and Q&A pods. Use the 4-arrow icon in the featured presentation pod to enter and exit the full screen view as you choose. To submit a comment or question for me or our presenter, use the Q&A pod and type in your question. I'll handle technical difficulties the best I can while hosting the webinar and interact with our presenter to answer your questions during the verbal Q&A period. Our speaker today is Salvador Ramirez. He's a Doctor of Plant Health student, UNL Agronomy and Horticulture at the University of Nebraska. Sal is working under Dr. Ray Driver and Dr. Virginia Jin, and a Doctor of Plant Health student and under Dr. Gary Hine. Sal completed his undergraduate degree at the University of California, Riverside. Under the guidance of Dr. Driver and Dr. Jin, Sal is characterizing the effect of 3 different agroecosystems in the state of Nebraska. 1, a crop rotation study with variable rates of nitrogen application. 2, an organic system in which he used cover crops to control weed species. And 3, what he will be presenting today, a corn production system in which corn stover was removed under 2 different tillage systems. And Sal has been doing an internship here at the Kellogg Soil Survey Lab where he's been doing a variety of laboratory analysis to learn all of those and the uniqueness of our Kellogg Soil Survey Lab and the products that we can provide. So Sal, I'm going to turn the presentation over to you.
Okay, and we can bring the lights here. My slides look pretty bad when the lights are on. Thank you for that kind introduction. So as was said earlier, my talk today is about the impact of corn stover removal under 2 different tillage systems on soil microbial communities and an irrigated continuous corn system in Nebraska. And I just want to highlight that this is done under the mentorship of Ray Driver and Virginia Jin, and in collaboration with Humberto Blanco, Lance Gunderson, and our technician, Dr. Elizabeth Jeske. So it's a very good group to work with. So just to provide an outline before we get started, today I'm going to be talking about who cares, specifically the benefits of corn stover and why some people want to use corn stover. I'll be talking about what we did, the field site, experimental design, soil sampling, fatty acid methyl extractions, which I will refer to now as the FAME and FAME nomenclature. What did we find? And there's a variety of things that we found, but for the interest of time, I'll only talk about microbial biomass and microbial community structure. And then finally, what do I think is going on? And for our first part of Who Cares, for those of you who are not familiar with what corn stover is everything that's left on the field after harvest, and it consists of residue, stalks, leaves, husk, and cobs. It's been wrongly referred to as trash. Some people refer to it as trash, but it has very many ecological benefits. Those include things like recycling plant nutrients, sequestering soil carbon, improving soil structure, enhancing activity of soil fauna, which is technically what we're going to talk about right now, controlling water runoff, and minimizing wind and water erosion. And when I provide this list of the benefits of what corn stover does, I want you to kind of appreciate the variety of things that fall within what we refer to now as soil health and soil quality. Right? We have chemical things, chemical benefits of corn stover, we have biological benefits, and we have soil physical benefits. And so one of our arguments today about what's going on with what we're doing with the soil is whether we're removing microbe homes or whether we're reducing microbe food, what people refer to as selective loss of niche, or if it's a general substrate limitation. And we'll get into that as to why we think what's going on with what we see. And so in terms of energy policies, people are interested in harvesting corn stover to use it in the production of second-generation biofuels, right? First-generation biofuels being biofuels made from corn grain. So this would be a second-generation biofuel made from corn stover. And this came all from the Biomass Research and Development Act of the year 2000, which created the Biomass Research and Development Technical Advisory Committee, which suggested that 5% of the nation's power, 20% of its transportation fuels, 25% of its chemicals would come from renewable resources by 2030. And this is what spurred this interest in using corn stover to make biofuel, because in 2003, the Department of Energy predicted that there was a need for more than approximately 1 billion dry tons of biomass feedstock, not only from corn stover, but just biomass in general, to reach this goal. So at that time, the question became, from where in the U.S., from what crop, and in what form will this biomass feedstock originate? And in 2000, the Union of Concerned Scientists released this diagram where they have the concentration of where biomass can come from. Not only corn stover, it could also be trash, wood chips, it could be all sorts of other things. But I want you to appreciate that a lot of that is concentrated, right? The darker the blue, the more that they're speculating can come from that part of the country. A lot of that would be coming from Nebraska, and that would be agricultural residues such as corn stover. And so in 2005, the billion-ton study identified specifically corn stover as an important biomass source. And in 2005, people were concerned that that would result in a decrease in soil organic carbon. Right? Our concerns have changed since then. And other people use corn stover to feed beef cattle. They use it for livestock bedding. And one of the things that I really haven't looked into, but some people use it or use it as mushroom compost. And so there are several reasons why people want to harvest this corn stover from a field. Right? There are economic reasons why farmers would want to remove some of this corn stover. What I'm going to be talking about today is the potential impacts of removing that corn stover from the field. So what did we do? We're going to talk about the field site, the experimental design, and the soil sampling. Our site is located in eastern Nebraska at the Eastern Nebraska Research and Extension Center, referred to as NREC now. If we zoom in on the site, I don't know how many of you have ever been out there. This is the farm where we have lunch and we meet and stuff. And then this is our site. And it's really cool because you can see what's been tilled and what's not, and I'll get to that in a bit. And so our experimental design, this is one block, is a split plot design with factorially assigned subplot treatments. So this subplot treatment consists of 3 levels of residue removal. One is all, and by all we mean about 70% because it's all that could be removed. One that's half, and with that I want you to see that 1 year is taken from one row and the next year is taken from the other row. That's why it's half. And then no residue removal, which is 0% residue removal. And then the whole plot treatment is a tillage treatment consisting of conventional disc tillage. And so we have the disc whole plot and the no-till whole plot and the factorially assigned subplot treatments. And we have 6 plots, right? Which you can kind of see from the aerial image, which I thought was pretty cool. We sampled at a depth of 8 inches after scraping the surface of the soil for corn residue. We didn't want to sample into the corn stover because that could throw off some of our measurements. 10 of the samples were taken between the rows, and 5 of the 15 were taken directly on the row, and they were all mixed up in a bucket, which we then used for our fame extraction. We sampled at 3 times of the corn growing season, which complicates our analysis. We sampled when corn, in 2014, we sampled when the corn was at V10-12, when it was at VTR1, and when it was at a reproductive stage of 6, and an earlier V5-E6 soil sampling for soil properties. But these are the sampling dates that we used to analyze stains. Right, we just used this, we sent the soil sample out to Ward, and we got the traditional soil test so that we can look at what's going on with that. And in 2015, we sampled when corn was at V89, VT, and R5. And I want you to appreciate that we're going to be comparing those dates. That V9 stage is comparable to that 2014 V10 stage. That VT stage is comparable to that VT R1 stage, and then these later reproductive stages, so that we wanted to also see how microbial biomass would change within the corn growing season. All right. So what exactly are FAMES? I've had a lot of people express interest on behalf of the USDA about FAMES and how it could be a potential thing that is measured. And so if we go back to high school biology, we recognize the lipid bilayer membrane of an organism, maybe a little transport protein, maybe some polysaccharides. What we focus on when we extract FAMES is this connection right here. So if we zoom in on that, if you remember your phosphate group and your glycerol group attached to your fatty acids, that's referred to as the hydrophobic head and the hydrophilic tail. right? And what we do in the lab is pretty simple. All we do is that we cleave this bond, the bond with the glycerol to the fatty acids, with methanol and potassium hydroxide, which then leaves those fatty acids in solution. And then we just measure those fatty acids. How those fatty acids are diagnostic of microbial groups, though, changes. So, for example, this fatty acid is 16 carbons long, right? Nothing special going on, no functional groups, no conjugation. So we refer to this as C16, and this is a general biomass marker which occurs in microorganisms. But we can get more complicated. For example, this is 16 carbons long as well, but there's a double bond, right? So we refer to this as C16, one double bond on the 11th carbon. This fatty acid occurs in arbuscular mycorrhizal fungi, right? And so that's how we can separate from that biomass and refer to this from now on as AMF. This fatty acid has a functional group, right? So this one's 18 carbons long, so you can guess that we call it C18 something. And this one's called 10-methyl because there's a methyl group on the 10th carbon of an 18th carbon long fatty acid. This fatty acid occurs in actinomycetes, which are bacteria that behave like fungi because they're filamentous, but we still like to keep track of them separately. So this is what we're extracting from soil to approximate soil microbial biomass. We have this list of fatty acids that we measure, right? So from now on, when I say total microbial biomass or fungal biomass, here's what I'm talking about. These red ones are bacterial biomass fanes. These green ones are actinomycete fanes, which we kind of lump in with the bacteria because they're bacteria. The cyclopropyl, these dark blue ones, they are also bacterial fanes, but they form a cyclic group in times of stress, according to the literature. This is the fungal biomarker. These yellow ones are just eukaryotes. This is a general biomarker, the first one I showed you, and this is AMF. So from now on, when we talk about biomass, when I partition out these fanes to approximate biomass, this is what I'm talking about. And we'll come back to this color coordination in a bit. So how did I statistically analyze what we saw? We used a repeated measures analysis of variance, and that's because if you remember, we measured 3 times within the corn growing season. So this means that we had to account for each of those measurements within the same observation. We used that repeated measures ANOVA for total microbial biomass, AMF, the ratio of fungal to bacterial biomass, fungal biomass, and bacterial biomass. That's what we use that ANOVA for. And then I used something called the canonical discriminant analysis, and I used that to determine soil microbial community structure. I won't show these results, once again, in the interest of time, but I also used several other soil parameters as covariates in an analysis of covariance. And then finally, I plan to use a multivariate analysis of variance when we get some enzyme data. And sometimes this comes up, so I like to be ready for that, but some people ask me for the model. And it's a simple response model to a mean and the effect of tillage, residue removal, time, the interaction of tillage and time, residue removal and time, and the 3-way interaction, which we never saw, the effect of block, and our error. Simple repeated measures ANOVA. So what did we find? So for this part of our results, I'm going to talk about biomass. So if we just look at total microbial biomass, we had a significant tillage by removal interaction in 2014, which is what this graph is showing you. From now on, I use the same colors that I used in the description of the experimental design. So green is none, this yellow is half, and this red is all. I was kind of hoping it stay in your mind like that. And everything that's hashed out like this is from the disk. So this graph is, once again, showing us this tillage interaction. And we see decreased total microbial biomass due to residue removal, but only in the no-till treatment, right? We don't see that decrease in total microbial biomass due to residue removal in the disk. Going on to 2015, we only have simple effects. We have the simple effect of tillage and the simple effect of removal, and we see that total microbial biomass is greater in the no-till than it is in the disk, but the same trend of decreasing total microbial biomass due to increasing residue removal. And this will be a common trend throughout each of the taxons that we look at. And actually, this is my favorite part of the story. This is the most interesting thing, that when we also looked at time, there's a significant tillage of sampling date. Remember when I showed you the 3 dates that we sampled in the corn growing season? Total microbial biomass within that season alone was greater when corn was at VTR1 compared to the earlier vegetative and reproductive stages for both years. right? So just within that season, this measurement was sensitive enough to see that change in total microbial biomass. So that was total microbial biomass. When we look at just arbuscular mycorrhizal fungi, not much going on in 2014 except for that sampling date. But in 2015, we had a significant main effect of tillage, where now we see AMF greater in the no-till, and a similar trend due to corn stage, where that greatest AMF biomass is when corn is at BTR1, and no difference from the R5 to the BT, but still we see those greater later on in the corn growing season. It's useful to look at the ratio of fungal biomass to bacterial biomass because fungi and bacteria have different functions in a soil, right? They occupy different environments of the soil, and they're responsible for different attributes of the soil. And so we like to see how wide that ratio is in response to treatments. And so the ratio of fungi to bacteria never responded to removal, but in both 2014 and 15, we saw a significant tillage effect and the same trend, right? That ratio was greater now in the disc. Right? So far, we've been seeing that microbial biomass or AMF biomass is greater in the no-till. But now we're seeing that this ratio is wider in the disc, and that threw me off, and I'll tell you why in a bit. And a really different trend in terms of just within season biomass, right? That ratio was greater when corn was at V89 as compared to the later stages. When we look at just saprophytic fungi alone, this is when we discovered that that ratio is being driven by fungal biomass because fungal biomass is greater in the disc. And this threw me off because when you disc, you're physically disrupting the soil environment. You're breaking up fungal hyphal networks. So I would have thought that we would have had a decrease in fungal biomass. I'll tell you why that's wrong in a bit. And then same trend as always. This is pretty consistent. As we increase the residue removal rate, we had a decrease in fungal biomass. Same exact trend in 2015. Same thing. And in terms of date, we saw that that ratio was driven once again by what's going on with the fungi because this is the same trend that we saw when we took the ratio of fungi to bacteria. And finally, for the total bacterial biomass ratio, or just for total bacterial, we have another interaction between tillage and removal where we see a more strong effect of residue removal within the no-till as opposed to the disc, and greater bacterial biomass in 2015 in the no-till. Right? So the bacterial biomass is greater in no-till, and the fungal biomass is greater in disc, but the same residue removal trend. And the same trend with the total microbial biomass when it comes to the corn growth stage effect of bacterial biomass. I'll come back to explaining why that happened. For our total microbial community structure, this is a canonical discriminant analysis, which forms linear combinations that explain the variance of a data set. And so this is the first significant canonical function, and this is the second significant canonical function. And this is a pretty beautiful graph because it clearly discriminates between the effect of no-till and disked and the all, pass, and none residue removal treatment. So our treatments are clearly changing our microbial community. And then when we specifically go to each fame, and this is why I color coordinated them the way I did in that other slide, we can see that they're color coordinating by color or by taxon, right? We have this red group of fames, and those are all bacterial. We have our fungal biomass over here, our cyclopropyls and our actinomycetes here. And if you just look at this graph, because I'm going to change the slide, just look at this graph, go on to 2015, we see that same grouping of microbial fames, suggesting that we are maintaining a constant microbial community structure, even though we are influencing due to our treatments of disking and residue removal, right? So what do we think is going on? And this is where I'm going to go back to, are we decreasing the amount of microbe food by taking residue off, or are we destroying the microbe homes by disking? A lot of people would like to argue that it is either or that one has a more strong effect than the other, but I think it's both all the time. We are both reducing their food, and we are both destroying their homes. And this is what I mean. When we have no-till and disc tillage, right? In this situation where we have residue removal decomposing at the soil surface, the decomposition products won't travel very far into the soil matrix. As opposed to when you have disc tillage, you not only chop those pieces up very small, you make them smaller, you incorporate them deeper into the soil profile. Now you've increased the surface area of that corn stover, and now it's deeper into the soil matrix where it's exposed to soil microbes, which is why we ended up seeing more fungal biomass than I expected. I really thought by disking we were going to just break that all up and they were not going to be able to come back. Instead, we ended up breaking them up, but we ended up feeding them with smaller stover particles deeper into the profile. When we look at just residue removal, to go back to these benefits of not removing corn stover, you can see that even though this is a chemical one, recycling of plant nutrients, and there could be an argument made for why that's mostly microbe food, right, general substrate limitation, we know that that influences soil structure. We know that that influences controlling water runoff. And so all of those things that are either decreasing the amount of substrates going into the soil still influence the microbe homes, macroaggregates, water infiltration, things of that nature. And so if we look at just soil moisture, we can see that there's a clear effect of residue removal and a tillage effect within the none treatment, suggesting that we are modifying the soil environment enough that the soil is holding water in a different way due to our treatments. And the same trend in 2015. And if we look at soil organic matter, which is probably argued as microbe food, right, a general substrate, we would see that we have decreasing soil organic matter measured on loss and ignition from that ward sample that we took, which this is pretty difficult to achieve, right? Soil organic matter isn't something that's so easily changed over time. We see that general loss of substrates, right? And same trend for 2015. So in summary, we had a decrease in total microbial biomass due to an increase in residue removal. AMF only responded to tillage in 2015. The ratio for fungi to bacterial biomass in both 2014 and 15 responded to tillage, but only greater in the disc compared to no-till. The fungi were greater in the disc and decreased with increasing residue removal, and bacterial biomass decreased with increasing residue removal in no-till in 2014. Right, suggesting both of them, suggesting general substrate limitation and suggesting loss of niche. And with that, I'd like to thank my advisor in our lab, the USDA, because these are USDA sites, my advisor for the Doctor of Plant Health program, and all the undergrads that helped us in the lab. This is Ray Driver, and this is our technician, Liz Jeske.
All right, well, thank you, Sal. We'll open it up for questions. Well, Sal, I think we've exhausted our questions online and in the room. I want to thank you for your time and effort to make this presentation. And this concludes our webinar presentation today.